In this video, PoshBee researchers from the Anses Sophia Antipolis lab show the procedure for pesticide residue analysis in nectar regurgitated by honeybees and bumblebees. PoshBee has many work packages. Here we will concentrate only on work package 2, which involves measuring chemical exposure, pathogens, and nutrition in honeybees, bumblebees, and solitary bees. In work package 1, field sites were selected to sample different matrices in a standardized way, according to a precise timetable. You will find more information on this particular design in a different short film. These different types of samples were intended for different laboratory analysis. These analyses are described in other short films. At the Anses Sophia Antipolis lab, we received many samples. Nectar regurgitated by honeybees and bumblebees for pesticide analysis, and honeybees, bumblebees, and solitary bees for pathogen analysis. In this short film, we will show how to proceed to analyze pesticide residues in nectar.
The nectar regurgitated by bees and bumblebees was collected and sent to the Anses Sophia Antipolis lab for pesticide analysis. When the sample of nectar arrives at the lab, its identification and the quantity of sample received is checked. Then, the sample of nectar is registered in order to have a unique identification number. And the sample is then stored in the freezer at -20 degrees Celsius until analysis. "87 pesticides, insecticides, fungicides, and herbicides are researched in nectar samples. The quantitative analysis of pesticide residues in nectar is performed using analytical techniques. The liquid chromatography and the gas chromatography, coupled with tandem mass spectrometry, named LC-MS-MS and GC-MS-MS respectively. Pesticide residues are identified and quantified by comparing the sample extract to a calibration standard solution and analyzing them by LC-MS-MS or GC-MS-MS according to the characteristics of the pesticides. Therefore, it is necessary to perform two kinds of sample preparation according to the analytical technique used for identification and quantification of pesticide residues. One for injection into LC-MS-MS and one for injection into GC-MS-MS. The sample is first prepared for analysis. The procedure of the sample preparation consists of the following steps. First of all, the sample is weighed and then homogenized on vortex. The Eppendorf tube is then centrifuged at 10,000 RPM for 5 minutes to obtain a limpid sample. A required quantity of the supernatant is taken for each analysis to be carried out. To prepare the different levels of the calibration curve, a blank matrix sugar solution is used, and different volumes of a multi-pesticide standard solution are added to this sugar solution. Finally, the sample to be analyzed is prepared. In all cases, calibration standard and nectar samples, the same concentration of internal standard is added in order to control the preparation of all samples. For the LC-MS-MS determination, the process consists to transfer the required quantity of nectar to a vial and mix the sample with internal standard, water and water miscible solvent. Then the vial is vortexed. Once mixing is complete, the sample is centrifuged at 500 rpm for 5 minutes. Then, the analyst installs the sample on the LC autosampler to be injected. For the GC-MS-MS determination, the process consists to transfer the required quantity of nectar to a vial and to add internal standard, water and water immiscible solvent to perform a liquid-liquid partition. The organic phase is collected with a specific pipette and is transferred to another vial. The extract is evaporated for dryness with the multi-vapor system. Then, the final extract is resuspended in an appropriate volume of organic solvent for GC analysis, and prior to the injection into GC-MS-MS, the sample is centrifuged. After preparing the sample, and prior to injection, the instrumental parameters of the chromatography system and the mass spectrometer are checked. Once LC-MS-MS and GC-MS-MS are ready, the analysis of the samples can begin. The different standards prepared in sugar solution used for the calibration curves, and the samples of nectar are placed into LC and GC autosamplers. At the start of the processing sequence, a robot arm takes a precise volume of sample to inject in the system. After injection into the system, the program is started. All the data required is transferred to the evaluation software and combined with the concentrations measured on the instrument. Calibration curves are built up for each of the 87 pesticides studied. Calculation is done automatically. This concludes the analysis.